Hello, Corey Barnum, Forensic Science, Forensic Service. Today I will be presenting adult forensic evidence, primarily SANE sexual assault forensic kit. We thank you for your participation in very important. Today we're going to talk about types of sexual assault and proper evidence packaging, review the contents of Oregon Sexual Assault Forensic Evidence Kit, which is used primarily for adults. We'll talk about different types of evidence transfer, and we'll finally conclude with some information about how evidence is tested from a serological standpoint at the crime lab. The Oregon State Police Forensic Services Division operates as one large laboratory system. There are 5 labs statewide that can all do the same type of biology processing analysis of safe kits and sexual assault evidence. Casework can be transferred between laboratories, but is typically worked by the laboratory nearest to where the incident occurred. This is to serve customer service and proximity to regional courthouses. The area shaded in red in the northwest corner of the state map is the counties, or are the counties, that are serviced by our Portland Metropolitan Area Crime Lab. The central, western, coastal counties are serviced by the Springfield Laboratory. The southwest counties in the state here are yellow on the map. Those are serviced by our Central Point Laboratory. The center and southern counties here in blue are serviced by the Bend Lab. And finally, the eastern counties and the northeastern counties, green on this map, are serviced by the Pendleton Laboratory. Physical evidence includes any items that are submitted by a law enforcement agency which may establish a crime has been committed, link a crime to a victim, and/or link a crime to a perpetrator. Biology processing is the discipline in the laboratory that processes physical evidence. In this discipline, we locate and characterize biological stains. From the information gained from that testing, we then collect samples of stains or target material to forward to the DNA section, and then our DNA unit can perform analysis on that collected sample. Sexual assault evidence can comprise any number of items of physical evidence. We commonly see safe or sexual assault forensic evidence kits. We may also receive penile swabbing forensic evidence kits or child safe kits. We also can examine underwear, the most common item of clothing that is submitted for sexual assault evidence. But any item that is worn as a garment can be examined. Condoms, bedding, and tampons and sanitary pads can also be examined. Proper evidence packaging and storage is dictated by that item's condition. Is it dry? Is it wet? Or is it liquid at the time of packaging? Everything that we advise you in regards to evidence packaging is in the intent of pursuing preserving that evidence in the most stable condition possible so that any evidence that might be present will not be damaged or deteriorate due to how that item was stored. Let's talk about dry evidence first. Dry items have either no stains or stains that are dried and stable. Often we see this on clothing, bedding, the exam paper that you may have had the patient undress on, or that they may have been examined on top of. Any trace evidence that you observe during the exam, apparent hairs, fibers, or miscellaneous debris, and fingernail scrapings would all fall under that category of dry evidence. Dry evidence should be placed in new, breathable packaging material, basically anything that is made of paper, a paper bag, or paper envelope. This allows for an air exchange between the evidence and the outside environment, which will help prevent mold or bacterial growth that we can see with plastic packaging. Seal with evidence tape and initial the seal when you have completed packaging a dry item in paper. It's important not to wet a glue seal with saliva or a moisture stick. Just use evidence tape to secure that envelope flap. Always label packaging with the patient's name, the location of collection. For example, if you've collected swabs, where on the patient's body those swabs were collected from, and what type of fluid may be expected, or the contact described by the patient. If the patient reports that the perpetrator kissed their neck and gave them a hickey, you would label that item with patient's neck, possible saliva, to help give the lab an idea of where that evidence was collected from and why. And dry evidence should be stored at room temperature, both during short term while you are completing the rest of the forensic examination, and also by the law enforcement agency. That's the most stable climate environment for this type of evidence. Wet evidence includes items that have wet stains or wet or damp at the time that you are working with them during the exam. Clothing that is wet on the patient's body, swabs that you've collected from the safe kit start out as wet items, tampons, pads, diapers, condoms, anything that has a wet stain that may have been collected from the patient's body or clothing. And finally, bedding, which is not typically encountered by a SANE nurse, but we do see this from law enforcement agencies. If possible, the proper handling of wet evidence is to air dry it first. So dry it completely, which will then get that evidence to a stable state for long-term storage, and then follow the procedure for dry evidence handling. If you have to package the item when wet, it's important to keep it cold by refrigeration or freezing and secure it until you transfer it to law enforcement personnel. You can place an item temporarily in plastic, but clearly label the condition of the item and the contents of the packaging. An example where you might have to do this is if you collect a tampon, a wet saturated tampon from the patient. You may have to place that item in plastic packaging in order to safely transfer it to law enforcement, but it needs to be taken out of that packaging, dried completely, and then stored in paper long-term by the law enforcement agency. One option is to place a tampon in a plastic specimen cup without putting a plastic lid over the top of that. That basically creates a tampon sauna, which is not good for evidence preservation. But if you place a tampon in a plastic evidence cup and then cover that with a paper towel or several bath tissues and tape around that paper, that creates a breathable lid on the container, and that entire container can now be placed inside a small brown paper bag and transferred to the law enforcement agency. In that way, you have kept the item from being a biohazard and potentially leaking out of the packaging, but you've also kept that air exchange so that it will dry out completely. Liquid evidence most commonly encountered during a SANE exam are blood alcohol and toxicology samples that are collected, so blood and urine. Proper handling of liquid evidence entails keeping it refrigerated and clearly marking the packaging refrigeration required so law enforcement is aware. Label the packaging with a biohazard label for your safety and anyone else's who is going to be handling that packaging. Please note that liquid evidence must be packaged separately from the safe kit due to differences in storage requirements and the potential glass hazard if you collected blood vials. Properly seal the evidence and label it as you would with other packaging. Patient's name, what was collected. Also note that blood and urine collected for blood alcohol concentration and toxicology are fine to be temporarily stored at room temperature. So if you've collected these samples and then you still have several hours left of your SANE exam, they will be fine on the counter in the room with you during that time. They do not immediately need to be refrigerated. This is something for longer term storage. So by the law enforcement agency, or if for some reason the law enforcement agency will not be able to collect these items until several hours after your SANE exam is completed, then you would want to secure these items in a refrigerator for the short term. All items that are not part of the actual safe kit, so that are not swab samples from the patient's body or hair samples from the patient's body, need to be packaged outside of the safe kit. Clothing, toxicology samples, tampons, condoms, anything that is an extraneous item collected from the patient's body needs to be packaged outside of the safe kit. Package it based on the condition, whether it's dry, wet, or liquid, and make sure to only put one item per container. If you collect multiple items of the patient's clothing, for example, each of those should go in a separate brown paper bag. Even though those items were all collected from the same patient, you don't want to put them in one bag because you may inadvertently cross-contaminate evidence from one item to another. Properly seal the item and add your initials to indicate the chain of custody, and then label that item. Patient's name, the contents, what is in the packaging, where it was collected from, date and time, and why it was collected. Is there possible semen, saliva, blood, or aggressive handling? Aggressive handling is skin-to-skin contact, and we'll talk about that in more detail in later slides. This photo is an example of the appropriate size brown paper bag for the size of the item that will go into it. When in doubt, use a larger package. It's possible that multiple scientists might have to open and then reseal the item within the lab, or the district attorney's office or multiple law enforcement representatives may have to open the packaging. So if you're not sure, use a larger bag. These photos all show the acceptable way to initial evidence tape seals. You want to sign your initials or your full name so that the writing is across both the tape and the packaging. So don't sign just within the tape. Make sure that part of that signature or your initials is actually continuing onto that envelope or brown paper bag. Let's talk about some of the contents for the adult female sexual assault forensic evidence kit. Depending on the described case scenario provided by the patient, collect the appropriate body swabs. If you are collecting internal body swabs, for example, oral, vaginal, cervical, and/or rectal, collect 4 swabs. If you are collecting external body swabs, for example, neck, thighs, and/or external genitalia, collect 2 swabs, 1 wet swab followed by 1 dry swab. When in doubt, and if the patient consents, collect swabs. The more information that you provide on that evidence packaging, the better. Letting the lab know where the swabs were collected from and why is very helpful for our evidence triage process. You can use unused envelopes from other kits to create a kit specific to the case scenario of the patient that you're working with at that time. You can also use an adult female kit and edit it to collect for a male patient. So feel free to cross out the evidence labels that are provided and handwrite in the applicable information for the exam that you're doing at that time. We need general location descriptions. These are examples of an oral swab standard that was submitted to the laboratory several years ago. And what you're seeing is information that is not helpful for the lab and was a lot of extra work for the SANE nurse. The nurse basically collected oral swab standards from several different locations within the patient's mouth and then labeled those specifically. We don't need that information. The DNA that is within someone's mouth is uniform across their entire mouth. So you don't need to take the time to do that, and it's not helpful. Labeling them oral is the right thing to do. The safe kit that we prefer to have collected and submitted to the Oregon State Police Forensic Services Division is this proprietary Oregon Sexual Assault Forensic Evidence, or SAFE kit. The lab works with the Sexual Assault Task Force and the SANE nurse program to improve and update these kits every couple of years. And so what you should see for every kit that comes through your facility is a barcode at the top with a multi-digit unique SAFE kit identifier number. Generally, the first 2 numbers are the year the kit was produced, and then the next numbers are the unique identification numbers for that kit. The most important piece of paperwork to complete and return inside the kit to the laboratory is what we call the FLIP, the Forensic laboratory information form. This is a one-page snapshot of the case scenario as provided by the patient. Important information for us on this form is the time elapsed between the incident and the sexual assault nurse examination, the areas of contact, and the types of contact. Please note if the patient does not know or is unable to provide a report of what specifically happened during the incident, it is very helpful for us that you write unknown here. That way we know that you considered the question, attempted to answer it, not that you just forgot or were unable to answer it. So if it's unknown, please tell us that and we'll proceed with that information. Possible areas of ejaculation are very helpful to know as well, because that can impact how that evidence is directed through the DNA analytical process. And the other piece of information that's very helpful is this here. What is the date and time of last voluntary sexual contact? Is there another individual or individuals who may also have contributed DNA to the patient but are not perpetrators in the incident? The adult female safe kit is divided into a set of swab envelopes and a set of hair and trace evidence envelopes. Let's talk about the swab envelopes first. We give you 4 envelopes that are labeled with the most common areas sampled. The first is for oral swabs. And as you may recall, we direct for internal areas that you collect 4 swabs. Please indicate if you're collecting these oral swabs for a DNA standard, or if you're collecting them because semen evidence may be present. We also ask that you collect 4 combined vaginal cervical swabs. Studies with consensual sexual partners have indicated that a swab sample that has collected both from the vaginal vault and the cervix is more likely to have recoverable male DNA than swabs that are collected just from the vaginal vault or just from the cervix. We also give you an external body swabs envelope and an other swabs envelope. So for both of these, indicate on the blank line where the swabs were collected from, for example, right breast, and why, possible saliva. Perhaps the patient tells you that the perpetrator performed penile and digital penetration of their anus. You would indicate anal for the location and then collect other aggressive handling, which is skin-to-skin contact, and possible semen due to the penile contact. Sometimes you may select all 3 types of contact if that is what is indicated by the patient's report, or you may not know. So if the patient is unable to provide a report or they have periods of memory lapse, you may want to collect breast swabs to preserve potential evidence. Then you would put breast swabs as the location and just write unknown on the envelope. And that way we know that that's why you collected those swabs. There's potentially evidence there, but you don't have a specific report. You may want to consider collecting hair evidence under some very specific criteria, which we will review in a future slide. If you do, we provide 3 pre-printed envelopes for pubic hair combings, pubic hair standards, and head hair standards. And we also include 1 comb to use if you are collecting pubic hair combings. If you do not collect any hair standards or combings, do not put the envelopes in the kit. We do not need empty envelopes. Only include them if you collect evidence. For adult male victims, we do have a male-specific kit called a penile swabbing forensic evidence kit. However, you can adapt an adult female kit for that purpose if that's all you have access to. Remember when you are working with a male patient to listen closely to what their report of the incident is. If they indicate that the rectal penetration from the patient, occurred by the perpetrator, you would collect rectal swabs for potential semen evidence from the perpetrator and ask the patient if they will submit their underwear for collection. There may be semen evidence from drainage on their underwear. If the perpetrator performed oral sex on the patient, you would collect penile swabs from the patient for possible saliva from the perpetrator and collect their underwear. It's possible that saliva transferred from their penis to their underwear while they were wearing it. If the patient was forced to perform oral sex on the perpetrator, collect oral swabs and potentially collect their shirt in case they may have spit up or had some sort of additional transfer of the semen from their mouth to the shirt they were wearing. Trace evidence includes apparent hairs, fibers, and miscellaneous debris. Most often, it is hair evidence that's encountered as trace evidence in a sexual assault case. If you are collecting standards, please remember that we need a fairly large number of hairs to represent an adequate standard. Approximately 24, both pulled and shed, either from all around the head or all over the pubic area. Cutting is not acceptable. We need that entire hair, including the root. So do not cut hairs for a hair standard. They have to be pulled and shed. If you're collecting pubic hair combings, we have the provided comb for you to comb through the pubic hair and then package that comb in the envelope with any trace evidence that is still adhering to it. You may also see miscellaneous trace items that just look out of place. For example, if you are examining the patient and you notice several blue fibers on her mons pubis. The easiest way to collect those, that is the least invasive to the patient and the best way to preserve the evidence, is to use a typical Post-it or adhesive note. There's just the right amount of adhesive on that Post-it to pick up that trace evidence without damaging it. And then you can fold that envelope, that Post-it note over onto itself so that the evidence is temporarily trapped and captured. Place that Post-it envelope and label it. So in this case, you would say fibers from patient's mons pubis. That way that evidence is preserved, and if we need to examine at the lab, we can. Do note that when you are collecting trace evidence, so apparent fibers or any hairs that you collected, you want to seal along the entire envelope opening. You don't have to seal the entire opening for swabs, but you need to do it for trace evidence so that nothing gets lost. If you do not collect head hair and pubic hair standards, it's a good idea to document that in your medical paperwork. Patient declined, or no indication of stranger to stranger assault. Something just so it's documented in your records why that evidence was not collected. If you are not familiar with the Sexual Assault Task Force's position paper on when to consider pubic hair standard collection, please go out to the SATF website. There are very specific criteria as to when it's recommended to consider this collection. If you have a stranger to stranger or unknown assailant, or if there are multiple assailants, if the patient has pubic hair and you note pubic hairs are collected in the pubic combing, or the assailant is an acquaintance that has not previously been in the environment where the assault occurred. If the incident meets any of those criteria, consider collecting pubic hair combing. This photo gives you an idea of why we ask for so many hairs in a pubic or head hair standard. If you recall from a couple of slides back, we ask for at least 24 pulled and shed hairs. The hairs that you see in this microscopic image were all taken from the same pubic hair standard. So as you can see, there is a lot of visual variation among hairs from the same individual from the same pubic environment. That is why we ask for so many if you are going to collect a standard. Moving on from the types of samples that you may collect, let's talk a little bit about what happens with the evidence once it's collected. Since spring of 2016, the OSP lab system has followed a direct to DNA process for safe kit evidence. That means that we no longer perform serological testing initially on safe kits. Our goal with this direct-to-DNA process is to obtain a faster turnaround so that we can get DNA results to the law enforcement agency and the district attorney's office in a timely fashion. We can still go back and perform serological testing on any safe kit if it's helpful to the investigation, but we don't do it routinely. If your patient would like to have a kit collected but wants to remain anonymous, it's important that you provide the patient's name on the forensic laboratory information form and/or the envelopes within the kit for verification purposes. Include the kit identifying number on the documentation that's given to the patient, and include the kit identifier on all additional packaging for items outside the kit, so those items can be linked back to each other. For the outer safe kit manila envelope, you can put anonymous for the patient's name so that their name is not identified on that outer packaging. Your documentation drives our analytical decisions. So the clarity of what you've collected, where you've collected it from, and why is really critical for us in making our triage decisions of what is going to go onto the DNA unit and what is going to be prioritized first to go to DNA. Aside from the safe kit, you may want to consider collecting additional evidence if the patient consents and if those items are available. In a typical sexual assault scenario, we don't usually need all of the clothing that the patient has reported to the exam wearing. However, it is helpful to collect the item that was closest to their skin, the most intimate layer they were wearing, for possible DNA and body fluid evidence transfer. So typically this is the underwear that the patient is wearing at the exam. You may collect other items of clothing if they are relevant. If the patient indicates that the perpetrator forced them to perform oral sex and then they spit up, it may be a good idea to collect the top that they were wearing because there may be semen evidence on that. If tampons or sanitary pads are present, if you encounter a condom within the vaginal vault, those items should be collected. If you see any trace evidence, anything that looks unusual or out of place on the victim's body or hair or on the undressing paper, you can collect that with an adhesive note, as described previously. Put that note in an envelope and then label what it was and why you collected it. If the patient reports scratching the suspect, consider collecting one wet followed by one dry swab from the fingers of that hand or both hands if they indicate that they used both. There are different ways that evidence can be transferred from one person or one item to another. Primary transfer is a direct transfer from one person to an item or a person. So if a suspect ejaculated on the victim's back, that semen being deposited on the victim's back is a primary transfer. If the suspect performed oral sex on the victim, their saliva to the victim's genitals is a primary transfer. Secondary transfer is when something has already been deposited and then it's transferred again. So if the suspect performs oral sex on the victim, we would expect the suspect's saliva to transfer to the victim's external genitalia. But then that saliva may also transfer to whatever clothing layer is closest, typically underwear. So it would be a good idea to collect the victim's underwear. We have seen cases where the suspect had oral contact to the victim's breast, and the breast swabs were negative for male DNA, but the bra swabs were positive. So when the patient put their bra on after the incident, the suspect's saliva was whipped from their skin onto the clothing. So consider collecting the victim's bra or shirt if they indicate that oral contact occurred to their chest from the perpetrator. Another type of secondary transfer is if the suspect vaginally penetrates the victim or patient with their penis, the victim's vaginal fluid on the suspect's penis may transfer to the suspect's underwear. So if that individual is apprehended in time, the law enforcement agency could collect their underwear. One of the phrases that I've mentioned a couple times during this presentation is what we at the lab call aggressive handling. This is skin-to-skin contact that is forceful and or prolonged. There's a significant enough time or force or both to transfer DNA from one person to another. The most common examples that we see in sexual assault cases are strangulation, grasping of the wrists or ankles to restrain or drag someone, manipulation of the breasts or buttocks, digital penetration, so fingers or hands within the vagina or rectum, and also inner thighs against outer thighs if both the suspect and the victim or patient have bared legs during this incident. If the patient reports aggressive handling to an area of the body, this is an external body location, so it's an example where you would collect 2 swabs, one wet, one dry, from the area the patient has described as being aggressively handled. DNA transfer is more likely to be present if no bathing has occurred and the contact occurred within 24 hours of the exam. But we want you to collect regardless. We will make the decision on whether that sample goes on for DNA analysis or not. Remember, when in doubt, collect. Touch contact is a totally different category of DNA transfer from a lab perspective. Touch is casual and limited handling of an area of the patient's body. It's not significant in time or in force, and so we would not expect enough DNA to be transferred for us to get a meaningful sample. For example, if the assailant removed the victim's clothing, there may or may not be an adequate DNA transfer. If the patient reports to you that that behavior occurred, consider collecting the clothing, but don't collect body swabs from those areas if there was only just the limited contact or brushing as the clothing was taken off. Once evidence comes into the laboratory for a sexual assault case, a couple of different things can happen. For a safe kit, we determine based on the case scenario provided by the police report, your medical paperwork, if it's provided with the kit, and the forensic laboratory information form, which are the appropriate body swabs to sample and send on to the DNA unit for analysis. If it's any other evidence that was collected for a safe kit, so underwear, tampon, other clothing, condom, anything, we follow a couple of different major steps. We'll locate the biological material. We'll characterize what it is, what those stains are through presumptive and confirmatory tests. We'll take a sample of the stain or biological material, and we'll send that sample on to the DNA unit for further analysis. So this is the serological analytical process. When we are screening items for semen, we are looking for 2 components, seminal fluid plus spermatozoa. We have 2 presumptive tests that we can perform, both of which are chemical tests that look for enzymes found in high concentrations in seminal fluid. Acid phosphatase, which is a non-specific enzyme found in seminal fluid and other body fluids, and prostate-specific antigen, which is a specific protein found in high concentrations in seminal fluid. For confirmatory testing, we do a microscopic screen to look for the presence of spermatozoa. This is an example of a pair of genes that were submitted for a case where a woman reported someone throwing semen on her pants. This is what the pants look like under normal lighting conditions. And this is what the pants look like using an alternate light source at 450 nanometers wavelength of light. This is a similar tool to a Woods lamp or another type of alternate light source that may be available through your hospital or clinic. And it's something that we use every day in the laboratory to try to characterize and target specific areas to test for possible body fluid staining. The example on your left is of a acid phosphatase test or AP test. Here in the clear fluid example, that's a negative reaction. That is what it looks like when the test chemicals are mixed together and there's nothing else there. It's just the test chemicals. This positive deep purple reaction that you see is when a small piece of fabric that has known seminal fluid on it is dropped into the test chemicals. You get that immediate billowing purple positive reaction. When we're testing for prostate-specific antigen, we use a commercially produced test card. It's a similar mechanism to a home pregnancy test, where we take a small fluid extract from the sample, and that is dropped into the sample well. The fluid wicks along that card, and if you get a line in the T or test region of the card, that is a positive result. Our confirmatory test for spermatozoa is a dye stain known as a Christmas tree stain, since it is a red followed by a green stain. That is applied to a smear made from an extract of a cutting from the evidence, either a swab or a piece of fabric. What you see here at 400 times magnification is a whole lot of spermatozoa. Every one of these little ruby pink colored cells is a human sperm cell head. This is a single human sperm cell head at 1,000 times magnification. We can also screen evidence for blood. We first do a visual examination looking for staining that looks like blood, red-brown staining, and then we can do a presumptive test called a phenolphthalein test that uses a chemical that detects the heme molecule in red blood cells. This swab, which has no color change, is negative. That represents just the test chemicals with no sample. This swab on the right with that bright pink color had blood staining present, then the test chemicals were applied, and that bold pink color came forward as the positive result. We can also screen evidence for saliva. Saliva may fluoresce or glow with an alternate light source. We also have a presumptive test, one of the commercially produced cards, similar to the prostate-specific antigen test. The test we use for saliva looks for the presence of amylase, which is an enzyme found as a normal constituent of human saliva. The amount of amylase can vary by individual, so this is not a confirmatory test. It's possible to obtain a negative test for amylase, but have saliva present in the sample. So we always have DNA analysis present as an option. Vaginal fluid may also fluoresce with an alternate light source, but we don't currently have any presumptive tests available to characterize a stain specifically as vaginal fluid. What we would do would be sample from a targeted area where vaginal fluid may be present and send that sample on for DNA analysis. The most common sources of DNA that we encounter in the laboratory are from blood, semen, saliva, vaginal fluid, perspiration, tissue, including skin cells, or hairs. We do not routinely examine urine or fecal material for DNA, as these two sources are very poor sources of DNA. And we perform nuclear DNA analysis, so testing DNA that has been extracted from the nuclei of cells. We do direct comparison from evidence samples to known standards. We can also use the CODIS database. CODIS stands for Combined DNA Index System. And there are two main sources of DNA information that we use for CODIS. Convicted offender profiles are entered for every state. And we also have forensic unknown or evidentiary profiles. Those two groups of DNA profiles are routinely and constantly compared against each other, which is how we're able to solve unknown cases. An individual will be convicted of a crime that compels them to provide a DNA standard. Once that individual's DNA is entered into the system, that's compared against unknown profiles from casework, and they can be matched and that person identified. We do have a way to provide feedback to you as SANE nurses, which is the Safe Kit Quality Assurance Evaluation Form. It's a way for us to provide feedback if we have suggestions for more clear evidence packaging or labeling, or perhaps if you have not followed guidelines. For example, if you collected many swabs from one body area and the ideal would have been 2, we can provide that feedback on the form. And we can also give you kudos and provide feedback on the fact that your documentation was very thorough and clear. This is an example of what the evaluation form looks like. It's just a couple of quick yes/no questions and then an option for us to provide comments as well. When these are completed, they're provided to the Sexual Assault Task Force SANE coordinator, and they are generally placed in your SANE file and are something that can be reviewed by the Certification Commission when your certification is renewed. If you need to order kits, you or your facility or organization, go to the Oregon State Police Forensic Services Division webpage, and you will see directions on how to order an adult safe kit. Our kits are currently put together by Relay Resources and are typically provided in boxes of 15. But updated instructions are available on the website. Our website also provides ordering information for child safe kits and penile swabbing forensic evidence kits. Those two kit types are made as requested directly by our Central Point laboratory. Finally, if you have any questions about the laboratory's piece of sexual assault evidence collection, or what you could have done during a SANE exam, or how to package some type of evidence, please feel free to call your local lab. The general numbers are provided here on this slide. They are also available on the Oregon State Police Forensic Services Division webpage. We are open generally during normal business hours. If it is after hours, contact the Oregon SANE Sexual Assault Examiner Coordinator for direction and questions. This concludes our certification training. Thank you so much for your time, and best of luck to you as you go through the process. Again, do not hesitate to call your local lab if you have questions in the future.